Chromatin immunoprecipitation followed by sequencing, or ChIP-seq, is a technique that can be used to discover the regulatory targets of transcription factors, histone modifications, and other DNA-associated proteins. In ChIP-seq, cells are harvested and DNA-binding protein is cross-linked to DNA in vivo with formaldehyde. Cells are lysed, releasing the cell contents, and chromatin is sheared into fragments of less than 1,000 base pairs, usually between 200 and 600 base pairs. DNA interacting with the target protein is immunoprecipitated using antibody and isolated by decross-linking. DNA-protein crosslinks are reversed and RNA and protein are digested. The DNA is purified, made into libraries, and sequenced. ChIP-seq data can also be used to find differential binding of transcription factors in different environmental conditions or cell types. Initially, ChIP was performed through hybridization on a microarray. However, ChIP sequencing has become the preferred method due to technological advancements, decreasing financial barriers to sequencing, and massive, high-quality data output. In this protocol, we will demonstrate techniques of performing ChIP-seq with bacterial biofilms, a major source of persistent and chronic infections. ChIP-seq will be performed on Salmonella Typhimurium biofilm and planktonic cells, targeting the master biofilm regulator, CsgD, to determine differential binding in the two cell types. In this video, we will demonstrate techniques of determining the appropriate amount of biofilm to harvest, normalizing to a planktonic control sample, homogenizing biofilm for cross-linker access, and performing routine ChIP-seq steps to obtain high-quality